As a kid, I always wanted a pet dragon. With today's technology, I can design one on my computer and hold a 3D printed version in my hand. But how can I make it real? Much like how a 3D printer uses a set of digital instructions to build my model, our bodies use biological instructions to build our cells, tissues, and limbs. Changing how the body forms starts with its DNA. The process by which a gene is expressed has two parts, transcription and translation. During transcription, DNA is unzipped. RNA nucleotides are bound to the template side of the DNA by an enzyme called RNA polymerase, forming a strand of messenger RNA. A ribosome then translates the messenger RNA, one codon at a time, and adds the corresponding amino acid to the growing chain. Once finished, this chain folds into a protein. A protein's function is a result of its shape, which is in turn determined by the order of amino acids that make it up. In this way, the A's, T's, C's, and G's of DNA directly correspond to the function of the final product. So how can I actually use this to make a dragon? Starting with the fertilized egg of an animal that already exists, say a lizard, I might be able to change the DNA to get my desired traits. Humans have actually been doing this for thousands of years in the form of selective breeding, which we can see in the great diversity of dog breeds we have today. The thing is, DNA only tells the body how to make certain proteins, not when and where they are needed. My dragon already has all the proteins it needs to make limbs, so there isn't really a gene we can just stick in and make wings grow on its back. This process is actually controlled by special proteins called transcription factors. They bind to the DNA at certain points, either helping mRNA polymerase or kicking it out. The presence of these factors controls whether a protein will be created and in what quantity. Animals like insects and arachnids can have many limbs because they use a pattern of segmented body parts. Each segment has a set of limbs which develop differently depending on where they are on the body. This is communicated to each cell using a transcription factor gradient. The factor is in high concentration on one side of the body and low concentration on the other. The amount of transcription factor in a cell tells it its position. Changing the concentration of these factors can trick a cell into thinking it's in a different position, with crazy results. This is the embryo of a lizard, specifically an anole. These little guys have become the model organism for reptile limb development and have had their entire genome sequenced. During early development, the beginnings of limbs form along the body. This is triggered by the presence of fibroblast growth factor, which is only produced in cells with the right range of fibroblast growth factor ligands. This process can be hijacked by inserting fibroblast growth factor directly into the area. If levels are maintained throughout early development, a limb can be grown anywhere on the body. Problem is, this limb will grow into a leg, not a wing. What limb will grow is controlled by a pair of transcription factor gradients, called T-box4 and T-box5. T-box4 causes hind limbs to form, while T-box5 causes front limbs. But no reptile today have wings, so we must look at another animal a bit further away on the evolutionary tree. Bats have wings which are anatomically very similar to the forelimbs of other mammals and even our lizard friend. They have a few key differences. The digits of bat wings are webbed, giving them a large surface to use for lift. All animal limbs start out this way, but the webbing is lost during development when the cells between the digits are programmed to die. Bats prevent this in their wings using the transcription factor MICE2. The other different thing bats have is extra long fingers on their front limbs. A 2010 study showed a correlation between the long digits of bats and the expression of 14 different transcription factors. The exact way each of these factors work is still a mystery, but researchers have shown that they are expressed in completely unique ways in bat wings, different than they are expressed in their legs or in any other animal tested. Transcription factors can be manipulated to make wild changes to organisms, such as growing a dragon or making a fly with legs for a face, but they can also be used for more practical purposes.
One of the most exciting of these prospects is using transcription factors to control targeted gene therapy treatments. By making a therapy gene require a transcription factor to work, when and where it will be expressed can be controlled using injections, and possibly even a simple pill. The idea is to avoid any possible side effects by making sure the patient creates the right amount of whatever gene product they need. While this branch of science is still extremely young, and it may take many years before we have the technology to really implement these theories, our growing understanding of the mechanisms of the very small has a potential for big things in the future. Who knows, maybe one day dragons will be as common as Pomeranians and teacup chihuahuas.